So I'm Trevor Pugh. I'm a senior scientist at the Princess Margaret Cancer Centre and director of genomics at the Ontario Institute for Cancer Research.
Hi there, I'm Daniel Schramek. I'm a junior investigator at the Lunenfeld-Tanenbaum Research Institute and an assistant professor at the University of Toronto.
So this study actually came out of a clinical trial we ran with Lily and Sue at the Princess Margaret Cancer Centre, a clinical trial called NETSEEK, where we wanted to understand genomic correlates with response to patients on that trial. And what we actually found from that study through analysis of tissue biopsy was a very distinct genomic signature. And it's exactly this genomic signature that we're now following up in the mouse model with our collaborator, Daniel Schramek.
So this research really started with the clinical findings Trevor had from patient samples showing these genomic alterations. And right now we're in a position of actually taking these alterations and asking in a mouse model, what do they actually entail within the mouse model, how these tumors look like, and can we find vulnerabilities in these mouse models to eventually go back into the patients and help the patients.
In the NETSEEK trial, we found a very striking genome signature in these tumors. Nearly half the genome was actually missing, and for these tumor cells to stay alive, they'd actually doubled their genome. So this is a very distinct genomic signature, and that's really why we wanted to build mouse models that would recapitulate what we found in patients. I think the benefits are going to be twofold. In our first study, we showed that PNETs develop through a very choreographed, stepwise progression of mutations and genomic changes. What we don't yet understand is how to disrupt that choreography therapeutically. So what drugs can act specifically on PNETs throughout their entire development? And that's really where this mouse model is so important.
And in terms of precision medicine, again, we want to figure out the subgroups of patients who might benefit from one treatment versus another treatment. And finding this out has a double advantage. One, we might be actually able to give the right treatment to the right people, and B, not give the right treatment to the wrong people. And on the other hand, save actually people from treatments which are unnecessary and still have an adverse effect for the patients themselves. So that's kind of where we are moving right now, and that's the goal of precision medicine as a whole.
And I can share, we've actually just were contacted over the weekend, someone already interested in our models who saw the project write up on the NETRF website and wanted to collaborate and already use those models before they're even fully developed. They're very excited about this project. Yeah, this project absolutely would not have happened without the support of NETRF. It was a really unusual opportunity to take what we found in patients and to immediately act on them to build a model back in mice from which we can then learn additional therapeutics. So thank you very much to NETRF, donors, and the patients who participated in those early studies. This study, especially in these very rare tumors, is very challenging to fund through other means. So thank you very much.